So thank you for the introduction and good morning. So I would like to start with what microRNA 122 is and how it affects the virus life cycle. So to start with, microRNA 122 is a microRNA that is only 22 nucleotides long, and it is abundantly found in our liver cells. And it has roles in lipid metabolism and also tumor suppression. And in terms of HCV, it is known to bind on the virus genome, and it is known to promote virus replication. So now I'll show you how exactly the microRNA 122 binds on the virus genome. So just within the first 47 nucleotides on the 5' UTR of the virus, there are binding sites for two copies of microRNA 122. And this microRNA 122 binding is required for the efficient replication of the virus. But how this occurs, or what is the mechanism behind this replication, is not exactly known. There are different speculations and different hypotheses proposed, and some have shown that that microRNA 122 stabilizes the genome in such a way that it promotes virus replication. The others have shown that it promotes translation of the virus in order to promote virus replication. And very few, along with us, have actually shown that microRNA 122 binding changes the virus genome structure in such a way that it promotes virus replication. But I'll come to the mechanisms of how it promotes replication in a while. But right now, I would like you to focus on how complicated this microRNA 122 binding is on the virus genome. So the microRNA binds to these two seed sites. It also has these accessory binding sites along with the 3' overhangs. And binding to the extreme 5' end along with the long overhang was shown to be essential in order to promote virus replication. But our question was, does HCV really require these complicated binding patterns in order to promote virus replication? Well, the answer is no. In fact, a single simple perfect match siRNA can also promote virus replication as long as its cleavage activity is lost. So, in our lab, we designed two siRNAs, Si18 to 36 and 19 to 37, and we tested them in a replication assay in which we used ago to knockout cells. So, these cells basically inhibit the cleavage activity of the siRNAs. And what we saw in this assay is that with endogenous microRNA 122, the level of replication of HCV is quite efficient. But when we antagonize MIR122 with an anti-MIR122, the level of replication decreases drastically. But to the same sample, when we add either of the two siRNAs, we see that the level of replication is rescued back to the original level, or it's even higher than that with Si19 to 37. So, this experiment was actually crucial to tell us that a single copy and a perfect match copy is enough to promote virus replication. And it does not also need to bind to the extreme 5' end, and also does not need the long overhangs to promote virus replication. What we also found with this experiment is that the siRNA sequences are not exactly same as the microRNA sequence, and the site where it binds is not exactly the same. So our next obvious question was, can siRNAs then bind anywhere on the genome to promote virus replication? So we designed multiple siRNAs that bound to first the 5' UTR of the genome, to the iris or the internal ribosomal entry site, which is required for the translation of the virus, and to the alternative predicted MIR122 binding sites in the NS5B or the 3' UTR regions. And what we saw was that most siRNAs that bound to the 5' UTR were able to promote virus replication, but none of those that bound to the other sites promoted replication. So I'll focus my talk on the ones that actually promoted replication. Um, Okay, so these are all the siRNAs that were designed, and we walked along each nucleotide in the 5' UTR. And what we saw was that most siRNAs colored in purple are the ones that promoted virus replication at different levels. And the most efficient siRNA that promoted replication was Si19 to 37. And as we moved, so it's right in the middle of this region. And as we moved away from this site, the level of replication started decreasing. And the region between which small RNAs could bind and promote replication was from nucleotide 13 to 44 on the 5' UTR. What we also found was that since we knew that siRNAs did not bind to the 5' end and did not also have the long overhangs, we wanted to see if having these two conditions fulfilled would enhance any replication. So what we did is we made an siRNA that bound to the 5' end, and we saw that there was an increase in the level of replication, and it was even higher than the most efficient siRNA that promoted replication. And when we added an siRNA that had these long overhangs, we saw that the level of replication actually decreased. So this means that binding to the 5' end actually promotes replication or enhances replication, but having the long overhang does not. So now that I've showed you that microRNA 122 promotes replication, and also we have these panel of siRNAs that can also promote replication, what is the exact mechanism behind this virus promotion? So we hypothesize that there's a structural change that happens when the small RNAs bind on the virus genome to promote replication. And we started by doing some prediction analysis with an online software. And what we saw was that when we used just HCV alone, we got a closed conformation structure. And these are the nucleotides where microRNAs are known to bind. And when we added either the microRNA 122 or SI19 to 37 in our prediction analysis, we now got an open conformation structure. And it actually formed another loop, which is a part of the internal ribosomal entry site. So it is required for the virus translation. So this was pretty exciting. But since these are just prediction analysis, we wanted to know what's going on in reality. And so we collaborated with Dr. Tushar Patel, who performs small angle X-ray scattering experiments. So these experiments basically give us low resolution structures of a macromolecule in a solution. And he basically did a size exclusion chromatography first to elude molecules on the basis of size. And what he found out was that there were peaks that were observed in case of when there was just HCV, and we had different peaks now when we added microRNA 122 to HCV. And when each peak was analyzed by Sachs to see what structure it gave, we had a 98 Armstrong molecule of size with HCV alone. And with HCV along with MIR122, we had higher sizes of molecules. And these were also different structures. So this was pretty exciting. And we would therefore like to do it along with our siRNAs to see if there are similarities in these structures. But see, since these are just like really preliminary data, we would also like to validate these data before moving forward. Also, because of the opportunity that was given to me by the CAN Hep C and TRR 179 internship exchange program, I was I could go and visit Dr. Volker Lohmann's lab, and along with Dr. Philip Schult, we were able to work on an objective, and that was to determine the time at which the HCV RNA can co-localize with microRNA 122 to understand when HCV actually co-localizes, when actually HCV requires microRNA 122 in its life cycle. So we started by doing fluorescence in situ hybridization in order to locate these RNAs in the cells and to actually detect these RNAs in the cells. So in green, if you see, these are the HCV RNA strands, and in red are the microRNA molecules. And co-localization is shown by the white arrows. So when HCV RNA was transfected into cells, we saw that co-localization was observed even from 4 hours till 48 hours in every time point. And when we did this with an infection of the HCV virus, we found out that co-localization was observed even at very early time points like 30 minutes and 60 minutes. So this suggested to us that microRNA 122 might be required throughout the life cycle of the virus. So in conclusion, I would like to say that siRNAs binding to nucleotide 1 to 3 and 13 to 44 can promote virus replication. The efficient virus replication was observed when siRNAs bind to 19 to 37, and it And as we move away from this region, the efficiency decreases. We also saw that with structured prediction analysis, there's a confirmation change from closed to open. And we could also see that with our SACS data, but we still have to validate this data. And with our co-localization data, we found that MIR122 co-localizes with HCV RNA as early as 30 minutes when we did infections, and as late as 48 hours when we did transfections. So yeah, thank you.
Okay, thank you so much for that. I have a quick question, or I guess more of a comment too as well. The SACS data that you're showing, you have do you have shape data for those constructs as well that you could do overlapping studies with, or
Right. So the shape data is not from our lab, but there are people who have done shape analysis on this. So we might be able to correlate. Actually, yeah, Silna Segan's lab has also done that data. So yeah.
Yeah, it'll be nice to see the overlay and see if the predicted structures from the different programs actually fit with the SACS data and the solution model.
Yeah, sure.
No, that's great.
Thanks.
It's exciting.
Great talk, Rasika. I was actually also curious about the SACS data. So I noticed in your HCV 5' UTR, you have like 3 peaks. Do you think those are corresponding to the different, more stable structures? And what do you think is the most stable structure?
I actually cannot comment on what like which structure would be more stable. I can comment on what we observed. So there were these peaks given to us, like the chromatogram given to us, and we saw the different peaks, and we had different structures and different sizes. This could be because of the different ratios in HCV to MIR122, so the stoichiometric difference, and this was also done just once. So we'll have to do it again to see if we have similar results, and to actually see which is the one that is with 2 copies of MIR122 or not. We'll have to do more experiments. Yeah.
Thank you. Very nice talk. Were there any differences, or do you know if there's any differences between the Hep C genotypes and the microRNA binding? Oh.
Okay, so as far as I know, the microRNA 122 binding region is conserved in most of the like genotypes, but I assume that the pattern of microRNA binding would be the same if the region is actually conserved between all genotypes.
I guess one of the limitations is the genotype of the functional virus that you can actually study. I mean, JFH1 is one of the few that you can actually work with, and to the degree that you're able to. So
right, exactly.
Probably limited in
itself. Yeah, that's what. Yeah, it's limited, right? Okay.
Thank you, Rasika. Just join me in thanking Rasika for that great talk.